Our first lecture in Unit 2 is going to deal with horizontal gene transfer in bacteria. Now, one of the characteristics of bacteria is how rapidly they can adapt to new environments, and that includes things like developing antibiotic resistance or picking up new virulence traits that help them in causing disease. And this ability to rapidly adapt is due to mutation and horizontal gene transfer. Now, the most important of these is horizontal gene transfer, as we'll see in this unit, because this allows for a more rapid adaptation where bacteria pick up a number of genes in one single transfer directly from another bacteria, as opposed to mutation, which is a slower process where single genes become modified during DNA replication. So let's look at our fundamental statements for this SoftChalk lesson. Mutation is a modification of gene function within bacteria, and it enables a bacteria to adapt to new environments, but it is a more slow process. Whereas horizontal gene transfer enables bacteria to respond and adapt to their environment much more rapidly by acquiring large DNA sequences from another bacteria in a single transfer. Horizontal gene transfer is a process in which organisms transfer genetic material to another organism that is not its offspring. Mechanisms of bacterial horizontal gene transfer include transformation, transduction, and conjugation. During transformation, a DNA fragment from a dead degraded bacterium, sometimes called environmental DNA, enters a competent recipient bacteria and is exchanged for a piece of DNA of the recipient. Now, this typically involves similar bacterial strains or strains of the same bacterial species. Transduction involves the transfer of either chromosomal DNA fragments or plasmid from one bacterium to another by a bacteriophage, a virus that only infects bacteria. Conjugation is a transfer of DNA from a living donor bacterium to a living recipient bacterium by cell-to-cell contact. In gram-negative bacteria, this typically involves a conjugation Pilis. A conjugative plasmid is self-transmissible. That is, it possesses conjugation genes, known as TRA genes, that enable the bacterium to form a mating pair with another organism, and origin of transfer sequences then determine where on the plasmid DNA transfer is initiated. Mobilizable plasmids that lack TRA genes for self-transmissibility can be co-transferred in a bacterium possessing a conjugative plasmid, which does code for the ability to make a mating pair. Transposons or jumping genes are small pieces of DNA that encode enzymes that enable the transposon to move or copy itself from one DNA location to another, either on the same molecule of DNA or a different molecule. Conjugative transposons carry the genes that enable mating pairs to form for conjugation. F+ conjugation is the transfer of an F+ plasmid possessing only TRA genes coding for a conjugation Pilis and mating pair formation from a donor bacterium to a recipient. But mobilizable plasmids may be co-transferred during F+ conjugation. And during HFR conjugation, an F+ plasmid with TRA genes coding for mating pair formation inserts into the bacterial chromosome to form an HFR bacterium. This results in a transfer of some chromosomal DNA from the donor to the recipient, which may be exchanged for a piece of the recipient's DNA through homologous recombination. And let's look at our detailed learning objectives for this SoftChalk lesson. Compare and contrast mutation and horizontal gene transfer as methods of enabling bacteria to respond to selective pressures and adapt to new environments. Define horizontal gene transfer and state the most common form of horizontal gene transfer in bacteria. Briefly describe the mechanism for transformation in bacteria. Briefly describe the following mechanisms of horizontal gene transfer in bacteria: generalized transduction and specialized transduction. Briefly describe the following mechanisms of horizontal gene transfer in bacteria: transfer of conjugative plasmids, conjugative transposons, and mobilizable plasmids in gram-negative bacteria, F+ conjugation, and HFR conjugation. And finally, describe R plasmids and the significance of R plasmids to medical microbiology. So those are our bullet points and the detailed learning objectives you'll need to know for the Unit 2 exam. So let's start out by comparing and contrasting a little bit about mutation and horizontal gene transfer in bacteria. Now we know that bacteria are able to respond to selective pressures and adapt to new environments by acquiring new genetic traits. And this is a result of mutation, mutation being a modification of a gene function in a bacterium, and as a result of horizontal gene transfer, which is the acquisition of new genes from other bacteria. Mutation occurs relatively slowly. The normal mutation rate in nature is around 1 out of every million to 1 out of every billion per nucleotide per bacterial generation. So mutation is relatively slow, but when bacterial populations are under stress, they can greatly increase their mutation rate to increase the chance of at least one of the mutant bacteria surviving whatever is causing stress. Now keep in mind, in mutation, a single gene is being modified. Several genes may be modified simultaneously with mutations on different locations, but a mutation is modifying a single gene function. As we saw when we talked about mutation, and if you want to review mutation, which you had on your take-home molecular genetics review exam, you can click on this box here and read again about substitution, addition, and deletion mutations and how that can affect the resulting gene. So mutation is modification typically of a single gene, but keep in mind most mutations are harmful to bacteria. Most mutations alter an existing gene, causing that gene to be non-functional. But occasionally a mutation in a gene results in the production of a new gene product, like a protein, that becomes an advantage to that bacterium, and then through selective pressure, that organism is selected for and can do better in that environment. Now, what we're going to really deal with more in this unit is horizontal gene transfer. And again, this enables the bacteria to respond and to adapt to their environment much more rapidly, because they're acquiring large DNA sequences, often a number of different genes, from another bacterium in a single transfer. So horizontal gene transfer we define as a process in which organisms transfer genetic material to another organism that is not its offspring. And the ability of bacteria and archaea to adapt to new environments as a part of bacterial evolution most frequently results from acquiring new genes through horizontal gene transfer rather than altering single gene functions through mutation. So horizontal gene transfer is going to cause a rather large-scale change in a bacterial genome. For example, virulence factors, often in combination, called pathogenicity islands, that are located on large, rather unstable parts of the bacterial genome, can be transferred to other bacteria by horizontal gene transfer. So virulence genes are the ones that code for the ability to cause harm or disease, and these can sometimes be transferred from one bacterium to another by horizontal gene transfer. And of course, the other prime thing we talk about is antibiotic resistance genes can also be transferred from one bacterium to another by horizontal gene transfer. So if these transferred genes provide an advantage, then they're selected for. If those transferred genes have no selective advantage, when the bacteria acquire them, they're eventually lost by deletion. There's a limited amount of space for DNA, including both plasmids and the chromosome inside a bacterium, so the genome has to remain relatively the same size over time. Now, there's three mechanisms of horizontal gene transfer we'll be looking at today: transformation, transduction, and conjugation. And the most common mechanism for horizontal gene transfer among bacteria is conjugation. Now, bacteria do acquire new genes through the other two mechanisms, transformation and transduction, but this is a little more rare transfer, and often it involves only the same species or closely related species. As we'll see, conjugation can occur between unrelated species. It's much more promiscuous in its transfer. Now, we gave you these three terms: transformation, transduction, and conjugation, in the materials you reviewed for your molecular genetics review exam, so you'd at least be familiar with the words when we got here. But now we're going to look at these processes in detail in this SoftChalk lesson. And there's a little self-check you can do on our introduction. So let's start with transformation. Now, transformation is a form of genetic recombination in which DNA fragments from a dead, degraded bacterium enter a competent recipient bacterium, and that donor DNA is exchanged for a piece of the recipient's DNA. But this usually involves what we call homologous recombination. And homologous recombination is a recombination of very similar DNA sequences that have almost the same nucleotide sequence. So the bacteria usually have to be very closely related to have these similar nucleotide sequences before transformation can occur. So this typically involves bacterial genes or strains of the same bacterial species. But there are a few bacteria, including some noted pathogens like Neisseria gonorrhoeae, Neisseria meningitis, Haemophilus influenzae, Legionella pneumophilus, Streptococcus pneumoniae, Helicobacter pylori, and others that are naturally competent and transformable. And by being competent, we mean that they're able to bind much more DNA than a non-competent bacterium. And in fact, some of the genera of bacteria that are naturally competent undergo autolysis, where the bacteria break apart just to provide DNA for homologous recombination. And some competent bacteria can kill non-competent ones in order to release their DNA to make it available for conjugation. But again, the key here is that during transformation, small DNA fragments, usually only about 10 genes long, and remember, a typical bacterium typically has about 3,000 genes. So these are pieces only about 10 genes long, are released from a dead, degraded bacterium. They bind to DNA binding proteins on the surface of a competent living recipient bacterium. And then either both strands of DNA or one strand of the DNA enter the recipient. The example we're going to show you shortly is where one strand enters, which is more common. That DNA fragment that from the donor that enters the recipient can then be exchanged for a piece of the recipient's DNA by special proteins called Rec-A proteins. And Rec stands for recombination. They allow DNA to recombine. And this involves a breakage and a reunion of paired DNA segments. And we see this in figure 1. So during the breakage and reunion process, a strand of the DNA, donor DNA, is broken with an endonuclease. Then some proteins bind to it so that it doesn't repair with the complementary strand. And when that is mixed with recipient DNA, a piece of the donor DNA shown in blue can cross over, like we see down here in step 4, and be inserted into the recipient's DNA, while a piece of the recipient's DNA in red can be bound to the donor DNA. So that's what's involved in breakage and reunion. And this is what allows the Rec-A proteins to eventually take a piece of donor DNA and insert it into the recipient's chromosome, like we see on the far left at the bottom. So the best way to learn these mechanisms, again, is to look at the animations, follow and visualize the process. It's a fairly common sense process when you visualize it. So here's our animation showing transformation in bacteria. So we have a donor bacterium here. We're showing its chromosome in red. And a recipient bacterium down here, we're showing its chromosome in blue. So during transformation, remember, the donor bacterium dies and is degraded. And as the bacterium is degraded, the DNA fragments. And those DNA fragments then go out into the environmental DNA. So sometimes we say that during transformation, bacteria pick up environmental DNA, these DNA fragments from dead donor bacterium. Now, also when that bacterium is degraded, plasmids can be released. And sometimes plasmids can then be transferred as well, as we'll see. So what happens next, we have a competent bacterium down here at the bottom, the recipient, and we have these donor fragments in the environment of the bacterium from the dead degraded bacterium. So here's one of our fragments, and in this illustration, that fragment of donor DNA is going to bind to special DNA binding proteins on the surface of the competent recipient bacterium, as we see here. Now, what often happens is that one strand of the donor DNA is degraded and the other strand is protected. And that one strand is what's going to eventually be inserted by the Rec-A proteins. So what's happening in this case is that a nuclease, shown in brown here, is going to degrade this orange strand. And then some single-stranded DNA binding proteins, the purple circles, are going to bind to and protect the red strand from the donor fragment. So there we see the orange strand being degraded and the red strand being protected by these single-stranded DNA binding proteins. Now the Rec-A proteins come into play. These Rec-A proteins, as we'll see here, can split the recipient bacterium's DNA and then insert that piece of recipient DNA into a piece of the red donor DNA into the blue recipient's DNA. The piece of the recipient's DNA that's chopped out or excised is eventually degraded because it's not being protected. So now these red genes from the dead donor bacterium has been inserted into one copy of the DNA in the competent recipient bacterium. And again, the excised strand can't replicate and is eventually degraded. But remember, although these DNA sequences are similar, the nucleotides along this red segment is pretty complementary to those around the light blue segment. They were similar orders of nucleotides where many of them can complementary base pair, but they can't all base pair because they're not identical. And bacteria have a very active DNA repair system that tries to correct the fact that some of the bases aren't lining up. So what the bacterium can do at this point is it can either cleave the DNA where you have the bases that don't quite match in red here and pair them with the existing light blue strand, and the bacterium would go back to its normal state. But it can also cleave the DNA on the light blue strand as not matching and make a complementary copy of the red strand, in which case then this trait would be a permanent trait of that bacterium. So if it were to cleave out the red bases because they don't quite match and make a complementary strand to the light blue strand, then notice that this has gone back to the original recipient DNA. That red strand from the donor has been lost. But there's an equal chance that instead of cleaving the DNA here and here to cleave out the red strand, because it's not matching, it can cleave on the complementary strand because it too isn't matching, like we see here. And now then it makes a complementary copy of that red strand, and this then now becomes a permanent trait in the recipient bacterium. That segment has permanently inserted into the chromosome of the recipient. So as you see, this is a little less common. It involves bacteria that have very similar DNA sequences, but it is a way in which genes from environmental DNA surrounding the bacterium can occasionally be inserted into a recipient's chromosome, if that is a competent recipient with DNA binding proteins on the surface. Now, our second method of horizontal gene transfer is called transduction. And transduction involves the transfer of DNA fragments from a donor bacterium to a recipient, but it's being carried by a bacteriophage, viruses that only infect bacteria. So anytime you hear transduction, that's going to involve bacteriophages as the tool for transferring donor DNA from one bacterium to a recipient bacterium. And there are two forms of transduction we're going to look at: generalized transduction and specialized transduction. Now, generalized transduction is the more common. This can occur either during the replication of lytic bacteriophages or temperate bacteriophages. Now, we're going to cover viruses and their life cycles, including the life cycles of bacteriophages, in Unit 4. But I'm going to review these life cycles just briefly here so you can understand a little better what's happening during transduction. Now, what happens during transduction is this is actually a mistake that occurs as the bacteriophage assembles after using the bacterium to make phage parts. Sometimes the capsid, which is the hollow protein shell that contains the genome of the bacteriophage, the virus, sometimes accidentally assembles around a small fragment of bacterial DNA rather than around a viral genome. It can also assemble around a plasmid sometimes instead of around a viral genome. And so when that bacterium, which is by mistake carrying a piece of bacterial DNA rather than bacteriophage DNA, when that virus injects the fragment of donor bacterial DNA into a recipient bacterium, it can be exchanged for a piece of the recipient's DNA, much like in transformation by homologous recombination. Now, to show you that process, let me just briefly review the lytic cycle of bacteriophages. That's in the link we see here. And again, we're going to take this up in detail in Unit 4. But you need to have just a rough idea of how bacteriophages, the viruses, replicate inside bacteria to understand how transduction works. So let's take a look at an animation from Unit 4 on the lytic cycle of lytic bacteriophages. And these are bacteriophages or viruses that infect bacterium, shut down the bacterium, use the bacterium's metabolic machinery to make viral parts, and the viral parts assemble, and then they lyse the bacteria to get out where they can then infect and replicate in subsequent bacteria. So the first step in the life cycle is called adsorption, where the bacteriophage has receptors on its surface that bind to receptors on the host bacterium. So this is the virus, the bacteriophage, looks kind of like an Apollo lunar landing module, adsorbing to the cell wall of a bacterium in this case. Once it adsorbs, then the virus injects its genome into the cytoplasm of the bacterium, as we see here. We call that penetration. So the viral genome, DNA or RNA, in this case it's DNA, enters the cytoplasm of the bacterium. During replication, showing here that viral genome that's been injected into the cytoplasm of the bacterium, replicates anywhere from maybe 50 to 200 times by complementary base pairing, makes copies of itself, using the bacterium's energy, nutrients, and such. Then the viral genome is transcribed into viral messenger RNA, coding for the various proteins and enzymes needed to actually make viruses from scratch inside the bacterium. Bacterial ribosomes attach to the viral messenger RNA and are translated into viral structural proteins and viral enzymes. So as you see, the viral parts are being made from scratch inside the host bacterium using the bacterium's metabolic machinery. Now, once the parts assemble, or once the parts are made, which is replication, then we have maturation, and that's where the parts assemble. So as we see in our maturation step here, here's the viral genome. I've only showed about half a dozen of these in here. But again, typically it'd be anywhere from 50 to 200 copies. And normally the bacteriophage parts that are being made assemble around the genome to form an intact virus or bacteriophage. So here we see the viruses assembling around the genome. And then during release, a lysozyme enzyme breaks down the peptidoglycan of the bacterium, causing it to burst from osmotic pressure, and the viruses or bacteriophages are dumped out. And at that point, they can infect and replicate in additional bacteria. So that's basically the steps involved in the lytic life cycle of bacteriophages. Now let's go back to our transduction, because the mistake is going to occur here that allows transduction during the maturation step. So let's look at our animation in horizontal gene transfer that just shows you generalized transduction. So if we go through the cycle again, it starts out with adsorption, where the bacteriophage adsorbs to receptors on a susceptible bacterium. And this is highly specific. A particular bacteriophage can typically only infect a specific bacterium. So there's the virus adsorbing. The virus then injects its genome, in this case DNA shown in green, into the cytoplasm of the bacterium. And at that point, enzymes coded for by the viral genome will begin to degrade the bacterial DNA to shut it down, so the virus can use the bacterium's metabolic machinery to turn the bacterium into a factory for making viruses. So there we see the bacterial chromosome being degraded. The virus is now using the bacterium's metabolic machinery to make viral parts. That was the replication we showed you. And then maturation is where the parts assemble around the viral genome. And this is the way it normally assembles. The capsid or protein shell that forms the head of the bacteriophage assembles around the viral genome. But every now and then, by mistake, instead of the capsid assembling around a viral genome, as we see on the far right here, it assembles around a piece of bacterial DNA. So that one virus, rather than carrying a viral genome, is carrying a fragment of the donor bacterial DNA. Now, it's also possible that this capsid could have formed around a plasmid if this bacterium had plasmids in it. But again, since the virus is still functional inside there, most of the viruses are normal, lysozyme breaks down the bacterial cell wall, and the viruses are released. But notice this one virus, shown by the red arrow, is now carrying bacterial DNA. The other ones are typical normal viruses that will enter new bacteria and replicate. But we're going to watch this one that's now carrying a piece of donor bacterial DNA by mistake. So when that virus infects a new recipient bacterium, shown as having a red chromosome, it absorbs and injects what it contains, but in this case, what it contains is not a viral genome, but a short piece of bacterial DNA. So now that blue piece of bacterial DNA from the donor enters the recipient bacterium. And at this point, it behaves like the fragments that enter during transformation, where Rec-A proteins can excise a piece of the recipient's chromosome and replace it with that little blue piece of bacterial DNA from the donor. And so now whatever these blue genes here code for from the donor now become a trait of the recipient. The piece of the recipient's DNA is eventually degraded. So now as the bacterium divides by binary fission, every daughter cell will now contain this little bit of bacterial DNA as part of its chromosome. So in generalized transduction, as we saw, it's a mistake that occurs during maturation or assembly, where a bacteriophage by mistake assembles around either a fragment of donor bacterial chromosomal DNA or around a plasmid. And then that one virus carries that donor bacteria DNA or plasmid to a recipient and injects it. And that becomes a trait of the recipient. Now, specialized transduction is a little more complex. This occurs during a different life cycle called the lysogenic life cycle of bacteriophages called temperate bacteriophages. And the difference here is that during with temperate bacteriophages, they can either enter a bacterium, shut it down and replicate like a lytic bacteriophage we just saw, or the viral genome can be inserted into the bacterial chromosome and become a part of the bacterial chromosome. And that's what we're going to follow here. During the lysogenic life cycle of temperate bacteriophages, where the viral DNA eventually inserts into the bacterium's DNA. Now, specialized transduction occurs during a rare case of the lysogenic life cycle. But before we look at that, with this animation down at the bottom, let's take a look at the lysogenic life cycle. Again, you don't have to know the details of this at this point, but you want to understand kind of what's happening here so we can see how specialized transduction is going to affect that life cycle. So again, with temperate bacteriophages, it begins the same as with the lytic bacteriophages, the ones that have a lytic life cycle. The bacteriophage absorbs receptors on the bacterial cell wall. The virus injects its genome into the cytoplasm of the bacterium. Now in the lytic cycle, or sometimes during the lysogenic cycle, the viral genome shuts down the bacterium, degrades its chromosome, and just replicates like it does in the lytic cycle. But what often happens instead with the lysogenic life cycle is that the viral genome is going to physically insert into the chromosome of the bacterium. And we call that prophage formation. So whenever bacteriophage genome inserts into the bacterium's chromosome, we say the virus is now a prophage. It's a part of the chromosome. And here we see the viral genome inserting into the chromosome of the bacterium. And again, as that bacterium divides by binary fission, every daughter bacterium is going to carry that prophage DNA. And that's normally how it remains. That's kind of the ultimate in pathogenicity for the virus. It never runs out of host because it becomes a part of the host. But in rare cases, maybe 1 out of every million to 1 out of every billion bacteria containing a prophage, a rare process called spontaneous induction occurs. And this is actually a result of mutation, as we'll see later on in Unit 4, where the genes that allow the prophage to remain in the chromosome mutate so they can no longer remain, and the virus can once again replicate. So what normally happens during spontaneous induction, which itself is a pretty rare event, is that the bacteriophage genome, the prophage here in black, come out of the bacterial chromosome. And at that point, they shut down the bacterium, they degrade its DNA, and they use the bacterium's metabolic machinery to replicate and to assemble, just like in the lytic cycle, and be released by lysis. So the rest of the cycle remains the same after that. But the point here is that if we look at spontaneous induction again, we'll see in a minute, that instead of the viral genome coming out where it's supposed to, here and here, the whole phage genome coming out of the bacterial chromosome, it comes out unevenly. Maybe it'll split here and here. So when that happens, as we'll see, a little bit of bacterial DNA becomes part of the viral genome, and part of the viral genome remains in the chromosome. So that's what we're going to look for during spontaneous induction. Rather than coming out as it's supposed to, it comes out in the wrong places, carrying a little bit of bacterial DNA with it. So let's look at that occurring in this animation, showing specialized transduction. So again, it starts out the same as with any bacteriophage. The bacteriophage absorbs to receptors on the bacterium. It injects its genome, shown here in green. But in this case, the temperate bacteriophage is going to become a prophage. It's going to insert its green genome into the blue chromosome of the bacterium and become a part of the bacterium's chromosome. So now, for all practical purposes, that viral genome, now called a prophage, is a part of the bacterium's chromosome. As the bacterium replicates by binary fission, every daughter bacterium is going to carry that prophage, and that's normally how it remains. But as we mentioned, in rare cases, 1 out of every million to 1 out of every billion bacteria with a prophage, spontaneous induction occurs. So here we see the bacterium dividing by binary fission. We see that every daughter cell is now carrying that prophage. But now let's look at the spontaneous induction. Now, as we mentioned when we showed you the lysogenic life cycle from Unit 4 a minute ago, normally during spontaneous induction, the viral genome would be excised right where it entered and come out intact. And keep in mind that spontaneous induction is a rare event, and then this mistake during spontaneous induction is a rare event of a rare event. So this doesn't occur as frequently as other means of horizontal gene transfer. Yet on a single Petri plate, you might see this happening 3 or 4 times because of how numerous the bacteria are. So now we're going to see that the viral genome comes out unevenly during specialized transduction. So notice when the viral genome was cut out of the bacterial chromosome, a little bit of the blue bacterial chromosome is still joined to most of the viral genome, but some of the viral genome in green was left in the chromosome. So again, instead of coming out here and here, it's going to cut out here and here, so that the prophage now has a little bit of bacterial DNA in it, but is missing some of its own genes because they're still part of the chromosome. Now, this virus can still replicate inside this bacterium because all the viral genes in green are in the same factory. So now, as the virus undergoes replication inside the bacterium using the bacterium's metabolic machinery, it shuts down the bacterium, but as the DNA of the virus replicates, the bacterial DNA replicates as a part of the viral genome. And so every virus is now going to carry that little piece of bacterial DNA inside its capsid. But remember, a little bit of the viral genome is left behind in the original bacterium. So these will not be complete viruses because they're missing some viral genes. But the point is that now every virus is carrying a little bit of bacterial DNA, shown in red here with the red arrow. So as the bacterium lyses and these viruses now infect new bacteria, they will absorb and inject what they're carrying, which is most of the viral genome and that little bit of blue bacterial DNA that was picked up by mistake. But now as the viral genome inserts into the recipient's chromosome to become a prophage, what's also being inserted is that little bit of blue bacterial DNA, which now becomes a part of the recipient's chromosome. And during binary fission, of course, every Donor Bacterium will carry that little piece of blue bacterial donor DNA. But no more viruses could be made in this bacterium because some of the green viral genes were left behind in the original host. So this is kind of a rare event of a rare event, but it does occur because bacteria have such a short generation time and are so very numerous. So on our scale, this is expressed with some frequency. And there's a little self-check you can do to review that. But the most common mechanism of horizontal gene transfer in bacteria is conjugation. And again, conjugation is a transfer of DNA from a living donor bacterium to a living recipient bacterium by cell-to-cell contact, formation of what's called a mating pair between two bacteria. And in gram-negative bacteria, that typically involves a conjugation or sex pillis. So conjugation is encoded by plasmids or transposons that we learned about in Unit 1. It involves a donor bacterium that contains a conjugative plasmid and a recipient cell that doesn't. Now, as we learned when we talked about plasmids in Unit 1, a conjugative plasmid is self-transmissible. It possesses the necessary genes for the plasmid to transmit itself to another bacterium through conjugation. And that's because of genes known as TRA genes or transfer genes. These are the genes that allow bacteria to form a mating pair and conjugate. So if the plasmid has these TRA genes, it is able to conjugate with other bacteria and form a mating pair, where two bacteria join together with a little bridge and DNA can be transferred from one bacterium to another. And the place where the DNA transfer begins is called the ORIT, or origin of transfer sequence. That determines where the DNA is split, and then one strand of the plasmid DNA is going to be transferred to the recipient. And we're going to show you an animation of that in a little bit, but this illustration kind of shows the general idea where this bacterium has, in addition to its chromosome or nucleoid, a conjugative plasmid which contains TRA genes as part of it. And that allows for one strand of the plasmid to enter a recipient. Now, we also learned about transposons or jumping genes back in Unit 1. Remember, these are small pieces of DNA that encode enzymes that enable the transposon to move or copy itself from one DNA location to another, either on the same molecule of DNA or on a different molecule. And again, these can be part of the bacterium's chromosome, in which case they're often called conjugative transposons because they wind up picking some TRA genes, or they can be part of plasmids. And again, they're typically about 1 to 12 genes long, a typical transposon in this case. But they can also contain genes coding for antibiotic resistance or virulence factors or other traits. So this animation here we looked at back in Unit 1 that shows how the transposons can be cut out of one piece of DNA and be inserted into another. And if you want to review that, just to show you how it can go from a plasmid to a chromosome, or from one part on a chromosome to another part, or from a chromosome to a plasmid, or from one plasmid to another plasmid, you can review that animation. But what we're looking at here specifically are conjugative transposons. Like conjugative plasmids, conjugative transposons carry genes that enable a mating pair to form, enabling conjugation. So this can allow both transposons themselves to be transmitted from one bacterium to another, or they can allow other mobilizable plasmids or mobilizable transposons to be transferred to a recipient, as long as one segment of DNA somewhere in that donor bacterium has TRA genes coding for a mating pair. Other plasmids and other transposons can also be transferred once the mating pair forms. But the key about conjugation that we want to keep stressing is that they possess a rather promiscuous transfer system that allows DNA to be transferred not only to like species, but to unrelated species or entirely different genera or family or orders of bacteria. So the ability of bacteria to rapidly adapt to new environments as a part of bacterial evolution often involves this acquisition of large DNA sequences located in conjugative transposons or conjugative plasmids from one bacterium to another through the process of conjugation. So let's look at this mechanism of conjugation with conjugative plasmids to start with, and we'll look at it in gram-negative since that's what we have our animation on. Now remember, most gram-negative can make pili, and so we see conjugation pili or sex pili in gram-negative bacteria. So this is going to involve a conjugation pilus on a donor bacterium binding to a recipient bacterium, typically one that lacks a conjugation pilus. As the pilus retracts or depolymerizes, it pulls the two bacteria together, and then other genes in the TRA segment can code for some membrane proteins that allow a bridge to form between the two bacteria to form a mating pair. And then a nuclease is going to break one strand of the plasmid at a specific site called the origin of transfer site. And that nicked broken strand then enters the recipient, but the other strand's not nicked and it remains behind in the donor. And then both donor and recipient plasmids make a complementary copy of themselves by normal complementary base pairing, adenine pairing with thymine, guanine with cytosine. And so when we're done, both bacteria now have a conjugative plasmid. So let's take a look at our animation here showing the transfer of both conjugative plasmids and then later mobilizable plasmids by conjugation in gram-negative bacteria. So here we have a donor bacterium. This is the chromosome on the left, which is not being transferred. And this is a conjugative plasmid. Now these green genes in the conjugative plasmid are the TRA genes, and these are the genes that allow it to form a conjugation pilus and eventually form a mating pair with another bacterium. The little gray segment is the origin of transfer. This is where one strand is going to be cut so it can enter a recipient. But then again, the plasmid may have additional genes like antibiotic resistance genes or a virulence gene shown here in red. So here's our bacterium with a conjugative plasmid. Because it has TRA genes, it can conjugate and form a mating pair. So the conjugation pilus attaches to a recipient bacterium. And as the pilus retracts or depolymerizes, that pulls the two bacteria together. And then other genes in the TRA segment here are going to code for various proteins that are going to allow a mating pair to form between the bacteria, or a little pore exists between the two bacteria. And at that point, it's called a mating pair. Now, again, these can be fairly unrelated bacteria. It doesn't have to be the same species or even the same genus. So a nuclease is going to split one strand of DNA at the origin of transfer, and that one strand of the plasmid is going to enter the recipient as we see here. Now notice the other strand of the plasmid showing in brown here is not cut and is not transferred. So now one strand of the plasmid is in the recipient shown in red, the other strand of the plasmid shown in brown is in the donor. Then each strand makes a complementary copy of itself. So now both bacteria contain that conjugative plasmid. Because they both now have TRA genes, both are able to conjugate and form mating pairs. And whatever these red genes coded for in the original plasmid are now found in both the donor and the recipient. So if those were antibiotic resistant genes, both bacteria would now have that antibiotic resistance, and both could now transfer that resistance to other bacteria through conjugation. So one thing that can happen with a conjugative plasmid is that that plasmid conjugative plasmid can be transferred from a donor to a recipient. But as long as one plasmid has these TRA genes, other plasmids can be co-transferred during that process. And these are called mobilizable plasmids. So this is a different plasmid from the conjugative plasmid. This mobilizable plasmid on the right doesn't have TRA genes. So if this plasmid was not in the bacterium, the conjugative plasmid, this bacterium couldn't form a mating pair. And this mobilizable plasmid couldn't be transmitted. But as long as these TRA genes in the conjugative plasmid are coding for mating pair formation, then once the mating pair forms, a nuclease can also nick a strand of the mobilizable plasmid, and that can be transmitted to a recipient. So as long as one of those plasmids has the TRA genes, as long as one of them is a conjugative plasmids, other plasmids the bacteria contain may also be transferred. And both of these could be co-transferred, but we're just showing the mobilizable plasmid being transferred here. But again, now one strand of the mobilizable plasmid is in the recipient, the other is still in the donor. Both make a complementary copy of themselves by complementary base pairing. So in this particular instance, this bacterium on the left still has a conjugative plasmid. It can still conjugate, and it also has a mobilizable plasmid. The one on the right only picked up the mobilizable plasmid. In this case, the conjugative plasmid wasn't transferred, so this one wouldn't be able to make a sex pilus and wouldn't be able to conjugate. But again, it's possible that a couple of different plasmid strands could be co-transferred. And this is a very common mechanism by which resistant plasmids, or R plasmids, which code for multiple antibiotic resistance and the formation of conjugation pilus, are transferred from a donor to a recipient. And this is a big problem, especially in treating gram-negative infections, especially many of the healthcare-associated infections like urinary tract infections, wound infections, pneumonia, septicemia, by common bacteria like E. coli, Proteus, Klebsiella, Enterobacter, Pseudomonas, and others. So this is the R plasmid conjugation as an example. And it looks just like the one we just looked at, except now these red genes are going to code for resistance to a number of different antibiotics. But again, this R plasmid has TRA genes and an origin of transfer sequence. And because it has TRA genes, it can form a conjugation pilus and a mating pair. So again, when this bacterium contacts another bacterium with its conjugation pilus, as the pilus retracts, the two bacteria are pulled close together. The TRA genes code for the completion of the mating pair. And then a nuclease is going to split one strand of the R plasmid so that that one strand will enter the recipient. But now remember, the other genes, shown in red and brown here, are the ones that actually code for resistance to a number of different antibiotics. Each strand makes a complementary copy of itself. And when all is done now, both bacteria contain R plasmids. Both can now conjugate with other bacteria and form mating pair and transfer those R plasmids onto other bacteria. And both are now multiple antibiotic resistance, because it also transferred the resistance genes. Now, F plus conjugation is a transfer specifically of an F plus plasmid. And this is a plasmid that has only TRA genes, coding for mating pair formation. Doesn't usually have any other additional genes. And so during conjugation, the F plus plasmid coding for the ability to conjugate is transferred from the donor to the recipient, and now both bacteria are capable of conjugating, but nothing else was transferred. And we see that occurring in this animation showing you F plus conjugation. So here we have the chromosome again on the left, and this is an F plus plasmid. Now these are all just TRA genes and an origin of transfer sequence. There's no additional genes being carried, but these TRA genes allow this bacterium to conjugate with others and form a mating pair. So again, the conjugation pilus makes contact with the recipient bacterium. As the conjugation pilus depolymerizes or retracts, a mating pair is formed between them. A nuclease at the ORIT sequence splits one strand of the F plus plasmid, allowing it to enter the recipient. The other strand of the F plus plasmid stays behind. Both bacteria make a complementary copy, and both bacteria are now able to form mating pairs, but notice no other genes were transferred. So it may seem, what would be the advantage of that, since nothing's being able to be transferred other than, in this case, than the conjugation pilus ability and the ability to make a mating pair. Well, keep in mind this bacteria may also contain mobilizable plasmids. So as long as there's an F plus plasmid in the bacterium, it can conjugate, but that means that other plasmids it carries, called mobilizable plasmids, could also be transferred during conjugation, as we see here. So again, once the mating pair forms, because the F plus plasmid coded for the ability to make a mating pair, a nuclease can also split a strand of another plasmid coding for other traits, and that could be transferred to the recipient. So both with conjugative transposons and conjugative plasmids, that allows some mobilizable plasmids or mobilizable transposons to be transferred. And again, both bacteria make a complementary copy of that mobilizable plasmid. And in this instance, the bacterium on the left, because it still has an F plus plasmid, can still conjugate. Now the one on the right didn't get the F plus plasmid, only the mobilizable plasmid, but it is possible both could be co-transferred given enough time. And the final form of conjugation is called HFR, or high frequency recombinant conjugation. And this is a rarer occurrence. This occurs when an F plus plasmid that has the TRA genes coding for mating pair formation actually inserts or integrates into the chromosome of the bacterium. So the F plus plasmid becomes a part of the chromosome. At that point, we call that an HFR bacterium. And this is one of the few cases where chromosomal DNA, part of the chromosome, can be transferred from one bacterium to another, rather than just transposons or conjugative plasmids. So again, let's look at the animation of this. So for HFR conjugation to occur, an F plus plasmid has to be able to insert into the chromosome of that donor bacterium, as we see happening here. So now we have an inserted F plus plasmid. Now because of the TRA genes, this bacterium can form a mating pair. So when this conjugates with the recipient and the Pilis attaches and retracts and the mating pair forms, then a nuclease will now actually split one strand of the chromosome, because the plasmid is inserted with the ORIT sequence as part of the chromosome. So as that is split with a nuclease, one strand of the chromosome can begin to enter the recipient. But remember that chromosome is very long. It could be 1,500 times longer than the bacterium. And because that mating pair bridge is rather fragile, and bacteria are always exhibiting Brownian motion and being bombarded by their environment, it's very possible that during this transfer, the two bacteria break apart. So it's very rare that a whole strand of a chromosome will enter a recipient during HFR conjugation. They usually break apart somewhere along the way. It does occur, but it takes almost 90 minutes for that to occur. And so for them to remain united as they're being hit by water molecules and jiggling about, they often do break apart. So often only a little bit of the chromosome enters the recipient. Now also notice that not all of the F plus plasmid enters the recipient, because where it's split, notice the ORT is in the middle of the plasmid here. So the trailing end of that chromosome has the remaining part of the F plus plasmid, the green shown right here. It only has part of an F plus plasmid, but it does have that piece of chromosomal DNA. So as mentioned, the mating pair usually breaks apart before the entire donor strand can pass into the recipient. The remaining strand of the chromosome that wasn't cut makes a complementary copy of itself, and the fragments that didn't get through are degraded. So this bacterium on the left still remains an HFR male. And the piece of chromosomal DNA in blue from the donor makes a complementary copy of itself. And at that point, like in transformation, that could insert into the red chromosome here of the recipient with Rec A proteins. And so whatever those blue genes from the donor bacterium on the left coded for, that now becomes a part of the recipient's chromosome. But the one on the right is not an HFR bacterium because it doesn't have a complete F plus plasmid. The one on the left is. So this is one of the few ways that chromosomal DNA, part of the chromosome, can be transferred from one bacterium to another during HFR conjugation. And there's a little review section on that. I want to back up just a little bit here where we talked about transposons, because since we've been looking at the conjugative plasmids. So remember that transposons or jumping genes can occasionally pick up TRA genes, in which case they're called conjugative transposons. That is, the transposon or jumping gene is also going to carry genes that code for the ability to form a mating pair and conjugate. So at that point, they're called conjugative transposons. So because the conjugative transposon allows a mating pair to form, these conjugative transposons can be transferred from one bacterium to another during conjugation. Mobilizable plasmids could also be transmitted if one of the transposons is a conjugative transposon code for a mating pair. And of course, even non-conjugative transposons can be transferred as long as one of them codes for a mating pair. So both conjugative plasmids and conjugative transposons allow for mating pairs to form, and that can allow for plasmids or transposons or both to be transmitted from one bacterium to another during conjugation. And again, with both conjugative plasmids and conjugative transposons, this is a rather promiscuous transfer of DNA. So the DNA is not just being transferred to like species or like strains like in transformation or transduction, but to unrelated species. And again, it's these conjugative plasmids and conjugative transposons that very rapidly allow bacteria to adapt to new environments as a part of their bacterial evolution. Because again, large DNA sequences are being directly transferred from one bacterium to another by conjugation. And finally, there's our self-quiz over the whole SoftChalk lesson on horizontal gene transfer that you can do. So that is horizontal gene transfer in bacteria.